In this lesson, we'll be looking at the stages in the life of a eukaryotic cell and how new copies of the cell are made when they're needed. The specification has the following to say about this. Aside from making new individuals, multicellular organisms grow larger and repair themselves by producing more cells. This process requires that new living material is made. Cell division is the process for making that new material. Chromosomes are found in the nucleus of eukaryotes. They're made up of DNA and they carry the instructions for making the proteins which determine the makeup of each cell, and therefore the entire organism. The instructions of the chromosomes are translated into new living material by the process of protein synthesis. In a cell that's not in the process of dividing, these chromosomes are so spread out within the nucleus that they can't be seen as individual structures. But when the cell enters the dividing stage, the chromosomes coil up and they condense and they become much shorter. And once this happens, the chromosomes can be seen as individual structures. The cells of each species have a characteristic number of chromosomes. One complete set comes from each parent, but the number of chromosomes doesn't relate to the extent of the evolution of the species or its complexity. Humans have got 23 chromosomes from each parent, making a total of 46. But cats have got 38 chromosomes in total, and dogs have got 78. Genes are the particular sections along the length of a chromosome that hold the instructions to make particular proteins. Each gene makes a different protein, but the majority of the DNA in human chromosomes doesn't function as part of a gene. There are large sections of DNA between the genes whose precise function is still being researched. The nucleus encloses the chromosomes. Now, it's the largest organelle in the cell, and they can be seen with a light microscope. In experiments where nuclei have been surgically removed from cells, the cells survive, but they have a very much reduced lifespan. No cell replication will take place, so although most of the reactions of life take place in the cytoplasm, the nucleus and the genetic material that it contains is needed to organize and direct those reactions and to bring about reproduction. In essence, the life cycle of a cell has 3 phases. In the first phase, called interphase, the cell grows by making more organelles. It may then go on to duplicate the DNA. So the single ladder-like strands of DNA become a double ladder-like strand in an X shape, with 2 identical copies along each side of the X-shaped chromosome. When attached to each other, the 2 strands are called sister chromatids. Together, or when they get separated, they're then called a chromosome. At this stage, the chromosomes coil up and appear as distinct granules in the nucleus, rather than just a single dark mass of threads called the chromatin. If it takes place, the next stage is mitosis, where the nucleus divides. The last phase, where the cytoplasm divides into two cells, follows on directly from mitosis. In mitosis, the nuclear membrane disappears and the chromosomes are pulled into single file along the center of the cell by microtubules. Next, the chromosomes are split apart, with each strand of the X-shaped chromosome being pulled to opposite sides of the cell. The nucleus membrane now forms around the two groups of single-stranded chromosomes. A cell membrane forms between the two sides. They separate, and the two cells have been formed, each with one strand of the previously X-shaped chromosome. The process of binary fission used by prokaryotes may be quicker and easier, but it's got no way to organize the multiple chromosomes found in eukaryotes. Mitosis produces genetically identical cells for eukaryotes, and this is vitally important for them. Creating regions full of identical specialist cells wouldn't be possible without it. Without regions of the body functioning as a specialist, the activities needed by the organism as a whole couldn't be carried out. A muscle couldn't contract to allow us to move. Some of the cells were hair or skin. A gland couldn't secrete enzymes if some of its cells were muscle cells. Producing identical cells is needed for growth and repair of the body. But they can also be useful for simpler eukaryotes as a form of reproduction. To recognize situations where mitosis has taken place, there are a few key things to remember. It's mostly about recognizing how it differs from meiosis. First one is to do with location. Mitosis is used to create identical copies of existing cells, which are then used for growth and for repair. Now, meiosis is only used to create the gametes used for sexual reproduction. That's the sperm and the egg cells in humans. So mitosis takes place throughout the body of an organism, but meiosis only takes place where the gametes are produced. In humans, that's the testes and the ovaries. The next thing to consider is what is produced. Now, mitosis produces diploid cells. The cell will start with two sets of chromosomes, and each of the daughter cells it creates will also have two sets of chromosomes. But meiosis also starts with a diploid cell with two sets of chromosomes, but then the daughter cells it produces are haploid, and they've only got one set of chromosomes. Another thing to think about are the number of divisions in the process. In mitosis, the cells divide only once. If the process starts with one cell and only two are produced, then it's mitosis. With meiosis, the cell divides twice. So a single cell will create 4 daughter cells, not just 2. The next thing to consider is a thing called synapsis, or also often called crossing over. In mitosis, the chromosomes line up at the center of the cell in single file. Now, in the first part of meiosis, the chromosomes line up next to each other, and because of that, they end up actually swapping sections of DNA with each other. Now, that doesn't happen in mitosis. That process of swapping sections and getting tangled up and swapping sections is called synapsis. The next thing is to do with sex. Gamete cells from meiosis fuse together in sexual reproduction. Now, if that happened with normal body cells, then the number of chromosomes in the resulting daughter cell would double each time it happened. Now, obviously, that couldn't be sustained. So, sexual reproduction relies on meiosis to produce the special gamete cells that have only got one set of chromosomes, which, when they join together, go back to the normal 2 sets, or diploid. So when a new individual is created using sexual reproduction, it's only the gametes that are created by meiosis. As a baby grows, it creates new cells using mitosis. So there's both meiosis and mitosis involved in a baby. Creating the gametes is with meiosis. Thereafter, everything is mitosis. Don't forget to download the resources for this video from the link in the description, especially the notes template. LearnFix has a lot more to offer than just the videos and the exam questions. The most popular aspect is the set of 12 online learning games we have for each topic. These help you practice your recall of the details. Now, when it comes to revision, LearnFix is there to help. After each section of the syllabus, there's a diagnostic quiz to help focus your revision efforts. You can then use a progress report to look back at the details for that particular topic, what went well and what needs more work. We're adding new features every month, so visit us at learnfix.com to find out more.